Hello everyone, welcome to the new session of cell cycle and cell division. Okay, so today we are going to solve the questions of the NCERT exercise from this chapter cell cycle and cell division. Okay, so let us start the session, and this is the first question: that what is the average cell cycle span for a mammalian cell? So they are asking the average cell cycle, how much time the cell cycle takes, okay, in a mammalian cell, like in a mammals. So the average cell cycle span for a mammalian cell is approximately 24 hours. So basically the cell cycle is having different phases, the interphase and the M phase. M phase can be mitosis phase in the all the body cells, right? So that is the somatic cells, and in that is the meiosis in the case of the germinal cells, right? So interphase and M phase. So interphase, if we see, then it is having different you can say the stages, like G1 as G2, okay, and there is one G0 also, but not in all the cells, right? So these are the different stages that comes in the cell cycle. And the average cell cycle, the duration of the average cell cycle span for the mammalian cell is approximately 24 hours. So in 24 hours, the cell cycle completes. Okay. Okay, moving to the next question. Distinguish cytokinesis and karyokinesis. So two types of divisions are there, cytokinesis and karyokinesis. So we have to tell about what is cytokinesis and what is karyokinesis. Okay, so cytokinesis. Cytokinesis is the biological process involving the division of cell cytoplasm during the mitosis or meiosis. Okay, so cytokinesis showing the division of the cytoplasm, and this comes after the karyokinesis, means after the division of the nucleus, right? So cytokinesis is the biological process involving the division of cell cytoplasm during the mitosis or meiosis. Whether it could be a mitosis or meiosis, but there is a cytokinesis division. There is a division of the cell cytoplasm. So that is called as cytokinesis. Okay. Now, what is karyokinesis then? So karyokinesis is the biological process involving the division of cells nucleus during the mitosis or meiosis. So this is the division of the nucleus. When nucleus divide its content equally, right, into the two nucleus, and now in a cell there are two nucleus, so that we can say that this is the karyokinesis process, and this occur in the mitosis as well as the meiosis, right? So the karyokinesis is the division of the nucleus, whereas the cytokinesis is the division of the cytoplasm, and that comes after the karyokinesis. Okay, next difference. Now the stages such as prophase, metaphase, anaphase and telophase are not present in the cytokinesis. It means that when the cytokinesis process is occurring, when the cytokinesis process is going on, so these are the certain stages that comes under the karyokinesis process. When the karyokinesis process is taking place, these stages we can see the prophase, we can see the metaphase, we can see the anaphase and the telophase, but these stages are totally absent in the case of cytokinesis. No such stages are seen. Okay, there is just a simple division of the cytoplasm. The cytoplasm divides equally into two halves, and there is like a constriction in the membrane and divide the cells into the two cells. Sometimes there is a cell plate formation also, as we have seen in the case of plant cell. So that helps in the division of the cytoplasm, that helps the division of the cell, right? But these stages are not seen at the time of cytokinesis. Now, in the case of karyokinesis, it is divided into 4 stages, right? When there is a karyokinesis process is going on, you can see these particular stages. What are those 4 stages? Prophase, metaphase, anaphase and telophase. These 4 stages you can clearly see, okay, under the microscope in the case of karyokinesis. Okay, moving to our next question. Describe the events taking place during the interphase. So interphase, there are so many stages. We have to discuss about the events that are taking place during the interphase. So what are the events? Okay, so there are different events, right? So the interphase involve a series of changes that prepare a cell for a division. So it adds, generally we understand that, or people think that interphase is a resting phase. Interphase, in the case of interphase, there is no such changes is going on in the cell. But this is not true. There are lot many changes are going in this particular stages. Okay, so interphase involves a series of changes that prepare a cell for a division. Actually, in this particular time, the cell is preparing for the division of the cell. Okay, so it is a period during which the cell experiences growth and DNA replication in an orderly manner, right? So there is a growth process. You can see the growth in the cell size enlarges, the content of the cell increases, the protoplasm increases. So this shows the growth, okay, growth of the cell. And also DNA replication, DNA get duplicated in an orderly manner. So interphase can be divided into 3 phases. So there are 3 different phases we can see in the case of interphase. So the first one is the G1 phase, and second is the S phase, and the third one is the G2 phase. So these are the 3 very important steps that we can divide the whole interphase, right? So the first one, G1 phase. What happens in the G1 phase actually? It is the stage during which the cell grows and prepare its DNA for the replication. Because the S phase, the very next phase, that is the S phase, in which the synthesis of the DNA takes place. DNA get duplicated. Replication of DNA is occurring in the S phase. So there are lot many enzymes required for the process of DNA replication. And we all know that the enzymes are not pre-prepared, or it is present in the cell. When it is required, then only it gets synthesized to conserve the energy. So what happened when the cell has to go for the replication in the S phase? So in the G1 phase, the cell synthesize all the enzymes, all the proteins that are required for the process of replication in the S phase. Right? So this is a very, very active phase. And in this phase, the cells is metabolically active, right? It is a very highly metabolically active. So it synthesizes all the proteins, all the enzymes that are required for the process of replication, because replication is just going to take place in the next stage. That is the S phase. So what happens in the S phase? It is the stage during which the DNA synthesis occur. Okay, so DNA gets synthesized here, DNA get duplicated here, replication of DNA occur in this S phase. Okay, so in this phase, the amount of DNA that is present per cell get doubles. The amount of DNA get doubles, but the chromosome number remains the same. The chromosome number has not been increased. It remains the same, right? But the content of the DNA get doubled. So this is the process of replication, and for this there is a requirement of many types of proteins. There is a requirement for many types of enzymes, right? So those enzymes, those proteins has been already get synthesized in the stage of G1 phase. Okay. So next comes the G2 phase. G2 phase. In this phase, the cell continues to grow and prepare itself for division. So the cells now it has to get enter into the M phase, whether it could be a mitosis phase or the meiosis phase, right? So for the mitosis and meiosis also, they require certain proteins, certain enzymes. So in the G2 phase, and okay, the cell continues to grow and prepare itself for the division. What is the preparation here? The preparation is to synthesize those enzymes and proteins, right? So the proteins and RNA required for the mitosis are synthesized during this particular stage, right? So here you can see the figure, okay? So G1, S and G2, and this all constitute to the interface. Now here you can see the G0 phase also. Sometimes the cells is in the, okay, not in any division phase, and no growth is also there. So the nerve cells, if you see, they are in the G0 phase, okay. And here you can see the metaphase. So in metaphase, prophase, metaphase, anaphase, telophase, okay, and then comes the cytokinesis process. So there is a karyokinesis and then there is a cytokinesis. So all these are the stages of the metaphase. Okay, metaphase can be divided into karyokinesis and cytokinesis, right? Okay, so here you can see it started from the one cell in the G1. So in the G1, the enzymes or the proteins that are required for the replication of DNA takes place in the G1 phase, and when the cell enter into the S phase, the replication process occurs. Now when the cell comes to the G2 phase, the proteins, the RNA, the enzymes that are required, okay, for the M phase is get synthesized into the G2 phase, right? Okay, so next question comes here. What is G0 or the quiescent phase of the cell cycle? What is G0 phase? Okay, so let us see. G0 or the quiescent phase is the stage wherein the cells remain metabolically active. Okay, all the metabolic activity is taking place inside the cell, but do not proliferate unless called to do so. The cells never proliferate. Proliferate means it divide. It do not get divided in this particular stages, right? So no proliferation is seen in those cells which are present in the G0 or the quiescent phase. Okay, so the quiescent phase is stage where the cells are metabolically active. Okay, metabolically active, but there is a no proliferation, okay, and unless called to do so. Okay, when sometimes what happens, the cells move out from the zero to the G1 phase. So at that time they shows the growth, they shows the synthesis or the preparation for the proliferation, right? Such cells are used for replacing the cell lost during the injury. Okay, sometimes there are some of the cells that are lost at the time of injury. So some of the cells divide and they goes to the, okay, to the G0 phase. So these are the such cells are used to replace the cells lost during the injury. Okay, so this is very important question. Moving to our next question. Why is the mitosis called equational division? Why the mitosis is called the equational division? So let us see the answer. Okay, so mitosis is the process of cell division wherein the chromosome replicate and get equally distributed into the two daughter cells, right? So mitosis is the process of the cell division. This is a type of cell division that occur in the somatic cells, mitosis, right? So the mitosis is the process of cell division where the chromosomes get replicated, chromosome divide, okay? It get replicated and get equally distributed into the two daughter cells. right? So the chromosomes that get replicated, half of the chromosome goes to one cell and the next half will goes to the another cells, right? So it is distributed equally into the two daughter cells. The chromosome number in each daughter cell is equal to the parent cell, and this is also taken care that the number of chromosomes that are present in the daughter cell is equal to that in the parent cell, and that is the diploid. It is diploid. Hence, the mitosis is known as equational division, and this mitosis is known as equational division, right? So this is very, very important, and this equational division is taking place in the mitosis, right? Moving to our next question, name the stages of the cell cycle at which one of the following events occur. So the events are here given, right? We have to tell the stage at which stage this event is occurring in the cell division. Okay, so the first one is here. Chromosomes are moved to the spindle equator. Second, centromere split and the chromatid separates. Third, pairing between the homologous chromosomes takes place, and the fourth one is the crossing over between the homologous chromosomes takes place. So let us see the answer. The first one is the chromosomes are moved to the spindle equator. So the equator are arranged where they are attached with the kinetochore, right, of the chromosome with the spindle fibers, and all the chromosomes are arranged on the equator. So this is occurring at the metaphase. Okay, this is occurring at the metaphase, right? Equator, spindle equators, right? And the second question, the centromere splits and the chromatid separate. So after the prophase, that comes the anaphase. So in the anaphase, the centromere splits and the chromatids get separated because each chromosome is having the two chromatids. So the chromatids get separated, right? And this is occurring at the anaphase. Okay, anaphase. Pairing between the homologous chromosomes takes place. So zygote of meiosis 1, okay, at the stage of meiosis 1, at this time of zygote, the pairing between the homologous chromosomes takes place. Homologous chromosomes comes and pair with it. Chromosome number 1, both are homologous. They comes and pair with it. Similarly, chromosome number 2, chromosome number 3, right? And this is occurring at the zygote of meiosis 1. Next, crossing over between the homologous chromosomes takes place. So when the homologous chromosomes comes together, they pair, then their arms, means their chromatids, makes a cross over it, and there is a breaking of these chromatids and the rejoining. So what happened at this time? The crossing over takes place. So this crossing over between the homologous chromosomes takes place at which stage? It takes place at the packaging of meiosis 1. Packaging of meiosis 1. Okay. Okay. Next question, describe the following: synapses, bivalence, and chiasmata, and draw a diagram to illustrate your answer, right? So with the help of diagram, you have to explain thus what is synapses, what is bivalent, and what is chiasmata. So let us see the answer. So synapses, right? The pairing of homologous chromosome is called as synapses, right? So the homologous chromosomes comes and pair, right? And this occurred during the second stage of prophase, that is the zygote. So in the prophase 1 or the zygote, what you see, the homologous chromosomes comes and pair with it, right? And there occur the synapse. Means the homologous chromosomes comes very close to each other, and you can see there is a closeness of it. Here you can see there is a gap, but there you can see there is a synapse of formation, right? So the pairing of homologous chromosome at this particular point, the pairing of the chromosomes has been taken place, and that is called as synapses. So this occur during the second stage of prophase or the zygote, right? That is the synapses. So synapses showing the pairing of the homologous chromosome in the figure. Now bivalent. So bivalent or the tetrad is a pair of synapsed homologous chromosomes. So they are forming during the zygotine stage of the prophase 1 of the meiosis. So bivalent. So you can see each chromosome has been opened and they are showing the two chromatids. So it is showing the bivalent, right? So the bivalent or tetrad. So if you see the whole pair of the chromosomes, right? So they are having two chromatid each, and total if you see, then there is a tetrad. Okay, bivalent or tetrad is a pair of synapsed homologous chromosome, synapsed homologous chromosomes, and they are formed during the zygotine stage of prophase 1 of meiosis, right? So this is formed during the zygotine stage of prophase 1 of meiosis. Okay, so this is very, very important to understand, right? So bivalent or the tetrad is showing the this is formed at the time of synapsed, okay, homologous chromosome formation. So next is the you can see four homologous chromatids, okay, one chromatid, two chromatid, three chromatid, and four chromatid in the two chromosomes. When the both the chromosomes get opened, okay, there are four chromatids. So four homologous chromatids or the two homologous chromosome. When you are talking about the chromosomes, then it is a bivalent, two homologous chromosome. But when you are talking about their opened chromatids, then there are four homologous chromatids. So then it is called as tetrad stage, right? Okay. Chiasmata. So chiasmata is the site where the two non-sister chromatids of the homologous chromosomes have crossed over, right? So here what happened? The non-sister chromatids of the homologous chromosome, okay, they cross each other. They not only the cross each other, but their segments can broken, and you can see the red color, this one is having the one blue color arm, and this blue color chromosome is having one red color arm. It means that there is a broken of the chromosomes, chromatids, and there is an exchange of the chromatid also, right? So chiasmata is the site where the two non-sister chromatids of homologous chromosomes have crossed over, and it represents the site of crossover, and it is also representing at this particular site the crossing over has been taken place. So it represents the site of crossover. It is formed during the diplotin stage of prophase 1 of meiosis. So at which stage we can see this? And this is seen in the diplotin stage of prophase 1 of meiosis. Okay, so chiasmata is showing the site where the crossing over has been taken place, right? So this is very, very important. Moving to our next question. How does the cytokinesis in plant cell differ from that of the animal cell? So there is a lot of difference between the cytokinesis in the plants and the animal cell. So what is the difference? So if we see the cytokinesis in plant cell and cytokinesis in animal cell in the tabular form, then in the plant cell, the division of the cytoplasm take place by the cell plate formation. Okay, so here the division of the cytoplasm is taking place by the cell plate formation. So at the site when the nucleus or the karyokinesis has been taken place, okay, so there is a cell plate formation in the cell, in the cytoplasm itself. So the division of the cytoplasm take place by the cell plate formation. Now How does it happens in the animal cell? The division of the cytoplasm take place by the cleavage. So there is a cleavage formation. This plasma membrane, it's forming the cleavage and moving inside this, okay, and reaches to the center, and then the cell get divided. So here the cleavage is responsible for the division of the cytoplasm, whereas in the case of plant cell, right, in the case of plant cell, the cell plate formation takes place. Cell plate formation takes place, and this divide the cytoplasm into the two. Now, the second difference, if we see, then the cell plate formation start at the center of the cell and grows toward the outward and toward the lateral walls, right? So from where, in the case of plant cell, from where the division of the cell is taking place, the division of the cell is taking place from the center of the cell, and this division is moving towards the outer, toward the lateral walls. So the division is started from the center inside the cell in the case of plant cell, and here in the case of animal cell, the cleavage start at the periphery and then moves inward, dividing the cell into the two parts. So the cleavage is started from the outside like this, and it moves inside. And when both the cleavage from upward and lower, it meets at the center, then the cell is divided into two. So there is a lot of difference between the cytokinesis in the plant cell and in the case of animal cell, right? So cell plate is there in the case of plant cell, whereas the cleavage process is there in the case of animal cell. Next question. Find examples where the four daughter cells from the meiosis are equal in size and where they are found unequal in size. So sometimes what happened at the meiosis process, there are four cells are formed in the meiosis process. Okay, so four daughter cells in the meiosis are equal in size, and there are at some point that you see that they are unequal in size. So if we see the spermatogenesis, that is the process of formation of sperm. Spermatogenesis, or the formation of sperm in the case of human beings, occur by the process of meiosis. So that is the gamete formation. Okay, the sperms are the gametes in the case of male, right? So spermatogenesis is the formation of a sperm in the human beings, and that occur by the process of meiosis division, and it results in the formation of four equal sized daughter cells. Very, very important, right? So after the meiosis division, there are four haploid cells are formed. Four haploid daughter cells are formed, and all the cells are formed here are equal in size. So this is one of the example you can say that shows the meiosis where the cells are formed, the new cells are formed, are of equal size. Now, the second type of meiosis we see in the case of oogenesis. Oogenesis, that is the formation of egg in the case of female. Okay, human female. So oogenesis, or the formation of ovum in human beings, occur by the process of meiosis here also. So again, this is the formation of ova, or the formation of egg is the process of meiosis, the gamete formation, right? So here also the meiosis division, the reduction division is taking place to forms the ova or to forms the egg. Now it results in the formation of four daughter cells which are unequal size. Now here first division that forms the egg and the bar body. Okay, it's a very small, the two cells, one of the cells is very, very small, whereas the other one is the larger one. Okay, now this larger one, okay, that divides again and it forms again one smaller and one larger. So at the last you can see there are three smaller whereas one larger. So this one larger is the functional one where the other three are non-functional. Okay, so here we can see the eggs that are formed by the process of meiosis. Okay, there is one single functional egg and that is a larger in size where the other are smaller in size. So meiosis division, if we see, then here we can see this is the example that is showing the unequal division of the cell, unequal in the division of the size of the cell. Here we are just taking the point, the size. Okay, in the case of spermatogenesis, all the four daughter cells are of equal size, whereas in the oogenesis process the cells are not in a equal size, right? This is the difference. Okay, next question. Distinguish anaphase of mitosis from the anaphase 1 of the meiosis. Okay, if we see the anaphase of mitosis division. So anaphase is the stage during which the centromere splits and the chromatids separate. Okay, the centromere gets splits into the two and the chromatids are get separated. So this is occur in the anaphase. The chromosomes move apart toward the opposite poles, and when this centromere splits, okay, and it is attached with the spindle fibers, so this spindle fibers moves in the opposite direction, right? So when it is joined with this centromere, it pushes the chromosome toward the opposite poles, right? So the chromosome move apart and move toward the opposite poles. Now these chromosomes are genetically identical. These are totally identical. Okay, and this is occurring in the anaphase of mitosis. Now, if we see in the case of anaphase 1 of meiosis, so during anaphase 1, the homologous chromosome gets separated, right? While the chromatids remain attached at their centromere. The homologous chromosomes are separated. They are not close to each other, the whole part of the chromosome. But yes, their centromeres are attached at the chromatids are still remain attached at the centromere region. Hence, in anaphase 1, the chromosomes of each bivalent pair separate while the sister chromatids remain together. Moving to our next question. List the main difference between the mitosis and meiosis. So there are number of divisions, okay, number of differences. So let us discuss with some of the differences. What is the difference between the mitosis and meiosis? So number one, in mitotic division, a single division result into the two daughter cells. Okay, single division is taking place and the cells is divided into the two daughter cells, that is the karyokinesis and cytokinesis, and simple, the cell divides into the two daughter cells of equal size. Now, in meiotic division involves the two successive division, meiosis 1 and meiosis 2. Now, these division results into the four daughter cells. So this is the major difference. Okay, mitosis gave rise to the two daughter cell, whereas the meiosis gave rise to the four daughter cell. Four daughter cells are formed at the end of the meiosis. This is because the daughter cells have the same diploid number of chromosome as the parent, right? So here the daughter cells, okay, it is a diploid cells. So daughter cells is having the same chromosome number. The two daughter cells which we have formed from the pre-existing cell by the process of mitosis division, it is having the same chromosome number from which cell they have divided. Okay, same chromosome number. Now, if we see the meiosis 1 is known as reduction division. Meiosis 1 is known as reduction division. And this is because the chromosome number is reduced to half. Okay, the chromosome number becomes half. If suppose the cells which has been undergone the meiosis division, it is having the 12 chromosome, then the new cells which has been formed, it will have 6 chromosomes. Okay, half of the chromosome number. So this is because the chromosome number is reduced to half. Meiosis 2 is known as equational division. Meiosis 1 is known as reduction division, whereas meiosis 2 is known as equational division. This is because the sister chromatids separate and the chromosome number remains the same, right? So this is the meiosis. Next difference, prophase is a short and does not comprise, okay, it is not having the different phases like pachytene, leptotene, zygotene, pachytene, diplotene, and dikinesis. These stages are not seen in the case of prophase of mitosis. Whereas in the case of meiosis, prophase 1 is a very long and comprises 5 phases. Okay, it is having the 5 phases. Okay, that is the leptotene, zygotene, pachytene, diplotene, and dikinesis. So all these stages we can see in the case of prophase 1, and it is having the 5 different phases. Next difference, there is no pairing of chromosomes crossing over or chiasmata formation during the prophase. Okay, there is a no pairing of the chromosomes, no crossing over or no chiasmata formation during the prophase. So it doesn't happen. Now in the zygotene stage of the prophase, the pairing of chromosomes occur. Okay, if we see the zygotene stage of the prophase, then the pairing of the chromosome occur. Now during the pachytene, the crossing over occurs. The chiasmata are formed in the diplotene stage. Synaptonemal complex is not formed. Okay, so in the case of mitosis, there is not so like a chiasmata formation or the synaptonemal complex formation or the crossing over formation taking place. So these structures are not formed in the case of mitosis. Whereas in the case of meiosis, okay, synaptonemal complex are formed during the zygotene stage of the prophase 1. So at the time of zygotene stage of the prophase 1, the synaptonemal complex are formed at the zygotene stage, right? Next difference, anaphase involve the separation of the chromatids of each chromosome. Okay, in the anaphase, so this involves the separation of the chromatids. The chromatids get separated from each other. So the separation of the chromatids of each other at anaphase. Now in the case of meiosis, it separates while the chromatids remain attached at their centromere. Okay, separation of the chromosomes occur of the chromatids while the chromatids remain attached to the centromere. During anaphase 2, the chromatids separate as a result of a splitting of the centromere. So at the time of anaphase 2, right, the division of the centromere takes place, and it divides the chromatids into the two parts. Last difference, mitosis plays a significant role in the healing, repair, and the growth of a cell. Right, so mitosis is a very, very important step that is taking place showing the significant role in the healing, repair, and the growth of the cell. Such type of division. Whereas meiosis bring about the variation and maintain the chromosome number from generation to generation. So such divisions taking place at the gamete formation, meiosis, right? So meiosis brings about the variation. It is bringing about the variation because you have seen there is a change of the chromosome by the process of DNA crossing over. Okay, at the time of crossing over, there is an exchange of genetic material. So both the chromosomes are now having the new recombination type. So meiosis brings about the variation. Now this new recombination, it's a very different term, right? And it maintains the chromosome number from the generation to generation. Next difference, what is the significance of meiosis? So what is the significance of meiosis? What is the use of the meiosis, right? So meiosis is the process involving the reduction in the amount of genetic material. So here there is a reduction in the amount of genetic material. It comprises the two successive nuclear and the cell division. Okay, so what does it comprises? It comprises the two successive stages, the nuclear division and the cell division, with a single cell cycle of the DNA replication that takes place in the interphase of the S phase. And as a result, at the end of meiosis 2, 4 haploid cells are formed. Now, significance. The meiosis maintain the chromosome number from generation to generation. If there is a reduction division, the chromosome number becomes half. And after the fertilization process, what you see? Each gamete is bringing the half chromosome number of the organism. So again, it is having the same chromosome number of that particular organism. So meiosis maintains the chromosome number from generation to generation, and it reduces the chromosome number to half, so that process of fertilization stores the original number in the zygote. So at the time of fertilization process, again, when two haploid gametes fuses together, okay, so the new cells will form, that is the zygote. It is having the same 2 chromosome number, right? Next is the variation are caused by the crossing over and the random distribution of the homologous chromosomes between the daughter cells. Variation play an important role in the evolution, right? So variation is a very, very important, and due to the crossing over, there is a mixing of the genes. There is a, you can say, the recombinant formation we can see in the case of this meiosis. Okay, so variations are caused by the crossover and the random distribution of the homologous chromosome between the daughter cells. Now, the variation play an important role in the evolution. Okay, variation is taking place, and this is one of the very important role in the evolution. Next difference, the chromosomal mutations are brought about by the induction of certain abnormalities. Okay, so chromosomal mutation, sometimes we see the mutation also, so change in the DNA sequence, change in the DNA number, right? So the chromosomal mutations are brought about by the individual with certain abnormalities, and these chromosomal mutations may be advantageous for an individual, but in some individuals these changes are very, very advantageous as well. Discuss with your teacher about haploid insects and the lower plants where the cell division occur, and second, some haploid cells in the higher plant where the cell divisions not occur. So here comes the answer. In some insects and the lower plant, fertilization is immediately followed by the zygotic meiosis. Okay, so there is a zygotic meiosis after the fertilization. Now, which leads to the production of haploid organism, right? So in some insect and some lower plant, after the fertilization, the zygote causes meiosis, which leads to the production of haploid organism. Now, this type of cycle is known as haplontic life cycle, where you see the haploid cells are formed. Okay, so this is the showing the haplontic life cycle, because some insects and some lower plants are, their plant body is haploid in number, right? So after fertilization, what happened? It becomes diploid. Now this diploid zygote undergo meiosis to form 4 haploid cells. So that is the haplontic life cycle. Now, the phenomena of polyploidy can be observed in some haploid cells in higher plants, which cells division does not occur, right? So polyploidy is a state in which the cell contains multiple pairs of the chromosomes than a basic set. Instead of basic set, what happened when there is a karyokinesis? So after the karyokinesis, right, there is a no cytoplasmic division. So polyploidy is a state in which the cell contain multiple pairs of the chromosome, okay, other than the basic set. So polyploidy can be artificially induced in plants by applying the colchicine to the cell's structure. So colchicine stops the cytokinesis process, and only there is a you can say the reduction division, okay. Next, can there be mitosis without DNA replication in the S phase? So mitotic cell division cannot take place without the DNA replication or the S phase. Okay, so this is a very important stage, that is the DNA replication in the S phase. So the two important events that take place during the S phase, what are the two important stages? One is the synthesis or the duplication of DNA, and other is the duplication of the centriole. Okay, so at two, what we see in the cell, okay, the nucleus, inside the nucleus, okay, the DNA gets doubled itself. That is the replication of the DNA or the duplication of the DNA. Now there is one another organelle that is called as centriole. So at the time of cell division, the centrioles duplicate itself. One move to the one pole and the second one moves to the another pole, right? So it is helping in the equational division. The DNA duplication is the important step as it maintains the chromosome number in the daughter cells. Okay, so this mitosis, that is the duplication, is important at the time of S phase, the DNA duplication. It is important to maintain the chromosome number in the daughter cells. And mitosis is in an equational phase. It is in an equational phase. So therefore the duplication of DNA is an important step. Okay, so this duplication is a very important step in the process of mitosis. Moving to our next question, can there be DNA replication without cell division? Okay, without cell division, can there be a DNA replication? There can be DNA replication where the without cell division. Now during the cell division, the parent cell get divided into two daughter cells. Okay, so what happened here? The cells first get divided into the two daughter cells. However, if there is a repeated replication of DNA without any cell division, then this DNA will keep accumulating inside the cell. Okay, so here every time what happened, the cell undergo replication, it divides, but it doesn't go for the cytokinesis process. No cell division takes place. Okay, only the DNA duplication is taking place, right? So this would increase the volume of the cell nucleus. Every time you can see the cell nucleus, okay, volume is enlarging, thereby causing cell explosion. So sometimes we see such type of changes. Okay, this would increase the volume of the cell nucleus and thereby causing the cell expansion. An example of DNA duplication without cell division is commonly observed in the salivary glands of Drosophila. Okay, so such type of DNA duplication without cell division, only the nucleus is duplicating every time, right? And that is observed in the salivary glands of the Drosophila. Now the chromosome undergoing repeated DNA duplication is known as polytene chromosome. Okay, but such type of division is called as polytene chromosome. Okay, moving to our next question, and that is the last one. Analyze the event during every stage of the cell cycle and notice how the following two parameters changes. The first one is the number of chromosome. Okay, how the number of chromosome per cell changes. Amount of DNA content C per cell changes. So during meiosis, the number of chromosome and the amount of DNA in a cell get changes. First, the number of chromosome N per cell. During anaphase 1, the meiotic cycle, the homologous chromosomes separate and start moving towards their respective folds. So what happened at the time of anaphase 1 in the meiotic cycle, the homologous chromosomes separate. They get separated and start moving towards the respective poles. And as a result, what happened? The bivalents get divided into two sister chromatids and receive half the chromosome present in the parent cell. Therefore, the number of the chromosome reduced in the anaphase 1 process, right? Now, second thing, amount of DNA content Okay, C per cell. What will be the amount of DNA content? During anaphase 2, the meiotic cell, the chromatids separate as a result of the splitting of the centromere. And it is the centromere that holds two sister chromatids at the chromosome of each chromosome. Now, as a result, the chromatids move towards their respective poles. The chromatids are moving towards their respective poles, and therefore, at each pole, a haploid number of chromosomes and a haploid amount of DNA are present. Half means half the chromosomes are present. Now, during mitosis, the number of chromosomes remains the same. There is no increase in the number of chromosomes. The DNA duplicate in the S phase and get separated in the two daughter cells during the anaphase. So though there is a duplication of DNA, right, that is in the S phase, but they get separated, the two daughter cells, during the anaphase. So as a result, the DNA content of the two newly formed daughter cells remains the same. Okay. It remains the same in the case of mitosis. But in the case of meiosis, we see that there is a half of the chromosome number is there. So the content of the chromosome is also half. So in this way, we have completed the solutions of the NCERT from the chapter cell cycle and cell division. Okay. So thank you very much for attending this session. We will meet again with some another session. Till then, take care.